we are going to discuss about an important topic which is referred to as vegetative propagation. Vegetative propagation of plants. So, what do you mean by vegetative propagation? The word vegetative refers to the parts of the plant body which may not be involved in the process of reproduction. So, when you take the vegetative parts of the plant body, it includes root, stem, and leaves. So, vegetative propagation is where new plants are obtained from the vegetative parts of the plant body, like root, stem, and leaves, without the involvement of the reproductive organ. So, which is the reproductive organ as far as flowering plants are concerned? It is the flower. So, without the involvement of flower, when the root, stem, and leaves are capable of giving rise to a new plant, it is termed as vegetative propagation. So, vegetative propagation is basically of two types. There are natural vegetative propagation happening, and we do exploit the property of vegetative propagation in artificial vegetative propagation as well. Artificially, we exploit the property of the plants to give rise to the new plant through their vegetative parts for industrial purpose, for economic purpose. We propagate those species artificially in the laboratory condition. When you take natural vegetative propagation, it is naturally occurring in certain plants where the root part of it can give rise to they would have storage roots where by axillary buds they would give rise to a new plant body. Like that, you have stem propagation as well, and you have propagation of new plant through their leaves as well. So these are all natural vegetative propagation methods. I mean, naturally certain plants are capable of giving rise to their new plant through their vegetative part. So these are certain vegetative propagation examples. I mean, say for example, when you have potato is a stem tuber. Okay, so they store food material in their roots, and they also would help in propagating. I mean, they propagate their species through their stem. Another example of vegetative propagation is onion, and the propagation method that is happening in strawberry is termed as runner, and ginger is an example of rhizome. So these are all vegetative natural vegetative propagation methods. And you have the propagation through their leaves, the example which is given in your ncrt book, which is the leaf of bryophyllum. You could find some axillary buds which are arising from the notches of those leaves, right? So these buds would fall off on maturity and would find a suitable substratum or the soil surface where it would grow as a new plant. So this is vegetative propagation through leaves, the natural vegetative propagation that is happening in leaves in the case of bryophyllum. So now it is about artificial vegetative propagation. First comes the process called stem cutting, wherein you remove a part of the stem with the nodal region. Node is the place where the leaf arises. With some nodes, you take the cutting small piece of the stem and you transfer it to a new pot. It is cutted and planted from the main plant into a soil. So this would give rise to branches, and this would form a new plant where the characteristic of it would be exactly like that of your parent plant. So this type of artificial vegetative propagation is termed as stem cutting, followed by layering. So this is called layering. Layering technique is where a portion of the stem will be bent like this and would be made to stand erect, or would be holding on properly to the deeper part of your soil. There would be a clip to hold it tightly to the soil surface. When it is like this for a period of time, it would start developing roots. So such a plant body would develop, a new plant would arise from the parent plant. So this main plant is termed as parent plant, and the one which is formed from the branch of it is the new plant. I mean, the plant which resembles by all means as that of the parent plant. So after it forming the root structures, it can be cut and detached from the parent body. Now you see, the new plant which is formed will have the characteristics exactly resembling that of your parent plant. So this technique is termed as layering, followed by grafting technique. Grafting technique is where you take a plant, where you take the root system of a plant, which is termed as stalk, and the shoot system of another plant, probably a large tree. A large trees are actually involved or would undergo this process of grafting. These technique are being followed for large trees. So you have a stalk, I mean the root system taken from one plant and the shoot system from yet another plant, which is termed as schizon. So now, to this stalk, this schizon is being grafted, and it would be wrapped with a cloth, and there will be wax that is applied onto the surface of it to cover both of the plants together, and it would be wrapped for a certain period of time, where they would start developing tissues in between, and they would grow into a new plant, where you can actually propagate many different types of larger trees with this grafting technique. So, coming to artificial vegetative propagation, first one is cutting, layering, and grafting. Cutting is where you remove a part of the stem with nodal region and you would transfer it to a new soil. Example: rose, money plant, sugarcane, and banana can be propagated by this technique, followed by layering. Layering is where you bend a stem region, a flexible stem portion of the main plant, and allow it to form root structures where it is digged into the soil, and that would develop into a new plant. This technique can be used to propagate jasmine, strawberry, raspberry, etc. Grafting is where it is done for larger trees like apple tree and pear tree. So the base portion, the root system, from where, I mean the plant where you take up the root system, it is termed as stalk, and the portion of the plant body where you take up the shoot system is termed as schizon. Next, we are going to see an important and an advanced method of artificial vegetative propagation, which is nothing but your plant tissue culture. So, plant tissue culture is a technique which involves the growth of plant in an protected condition, or you otherwise call it as in vitro culturing or in vitro growth condition. So, it is where you grow these plants from a piece of the plant tissue which is taken from the parent plant, and it would be grown in a laboratory condition. That is what we refer it to as in vitro culturing. Okay, so you take a piece of the leaf from the plant body, from where a small portion of it is taken and the tissue is actually isolated. So now this isolated cells or a portion of the plant can be taken from where the cells are collected. So the cells would be then transferred to the growth medium. So this growth medium will be supplied or supplemented with all sort of nutrients and required hormones for the growth and development of the plant body. So this is referred to as growth medium. So in this growth medium, the cells would grow and form a group of undifferentiated cell, which is termed as callus. That would be the initial mass of cells that are formed in the growth medium. Later, when this callus is properly induced for differentiation, it would give rise to small plantlets. This plantlets would start developing root system and shoot system. Then it would be transferred to a pot condition, which where it would be allowed to grow and further develop. So this technique is termed as plant tissue culture. So, how do you define plant tissue culture? It is also called as micropropagation, where we take only some portion of the plant tissue and we are inducing the growth of the new plant. So, the production of new plant from a small piece of the plant tissue that is taken up from the plant, I mean, it should be taken from the suitable growing tip of the plant body so that it can be induced or transferred properly into a growth medium and allowed to grow. So there are certain plants, especially ornamental plants such as orchids and dahlia, are propagated by this micropropagation or plant tissue culture technique.
We have come to the end of this video. Thanks for watching BioPhobia. Kindly do comment below if you have any doubts or clarifications regarding this session. Thank you once again.